Hello, welcome. Welcome everyone to Frontiers of Brain Health, where we get to invite the most amazing scientists every week to share with you the leading edge scientific discoveries that are coming out in our world today. My name is Dr. Sandy Bond Chapman. I'm the founder and chief director of the Center for Brain Health, and I've dedicated my life to figuring out new ways to measure the upward potential of the human mind versus what's wrong with your brain, which 99% of our measures do, and figuring out how can we enhance brain health starting young and going every day of our life. We will have a few minutes to answer some questions, and I'd love, Ted, I hope you're going to leave time for questions at the end. We usually have double the number of people online, so I want to leave time for you. Use the Q&A choice function in your computer, and then we'll have time for all of you here that took time to be with us. For those of you that don't know much about the Center for Brain Health, I hope this is just the first of your journey to find out everything you can do to make your brain healthier, because never has a moonshot to optimize brain health been more urgent or more possible because of the cutting edge science that's coming out here, as well as our collaborators with universities all over the world to make brain health a household item. Now, I have the distinct pleasure and honor to introduce my colleague, my friend, Theodore, or Ted, as we call him, Price, who is the Ashbel Smith Professor in the Department of Neuroscience at the University of Texas at Dallas. He is the director of the Center for Advanced Pain Studies. I bet if I were to ask you today how many of you have pain, most of us would raise our hand. He got his PhD from Ken Hargreaves at UT Health San Antonio, and he was one of the most cited professors in pharmacology of pain and inflammation. And then he got his postdoc from Fernando Cervello at McGill, who was the chief editor of Neurobiology of Pain. And now that role is held by Dr. Ted Price. Ted started his independent lab in 2007 at the University of Arizona, and we highly recruited him. I still remember our dean getting so excited about the chance to recruit Dr. Ted Price to UT Dallas. He rapidly expanded his lab's goals to identify molecular mechanisms causing chronic pain, with an emphasis on developing new drugs to treat pain. It's one thing to understand pain. We want someone to get rid of pain. His lab focuses on the human molecular neuroscience, focused on specialization of the dorsal root ganglion and the spinal dorsal horn. He's published more than 200 peer-reviewed studies. He continues to be one of the most successfully funded pain scientists in the country, with NIH funding for 15 years. He's co-founder of many companies. You wonder where he has time to do fun, but I think he should also be awarded the best dressed neuroscientist in any gathering, you will notice. And his favorite hobby is basketball. So he's a little bit in mourning because of our loss of Luca. But please join me in welcoming Dr. Ted Price.
I'm more than in mourning about Luca. It's the worst thing ever. All right, so thank you very much, Sandy, for the really nice introduction. I want to start by saying that my mom is here, so thanks for coming, Mom. Yeah, not every day I get to give a talk in front of my mom. So what I'm going to do today is tell you about the work that my lab is doing and the work that's happening in our center, and we're very excited about also expanding the work in our center to be working with Sandy's center too, so that's a good opportunity that should come from today. So, I'm interested in trying to develop new therapeutics for pain, and one of the things that I should say about kind of my scientific journey is that I pretty much have changed over the last 10 years when I came to UTD. It's actually been 11 years now. changed completely what we do in the lab. So when I did my PhD and my postdoc, I was working with animal models of pain, and when I was at the University of Arizona, I also did animal model work. And Sandy just described what our work does as human molecular neuroscience. So we pretty much totally transformed our work from doing animal models to now working with humans and human tissues. So what I want to start with is explain why we made that decision. So there's lots of reasons to study the human nervous system. But my reason for doing that is a little bit different than some others, I think. So I did my PhD in pharmacology because I wanted to be involved in drug development for neurological disorders. I didn't really know I wanted to work on pain when I was doing this, but I had an amazing opportunity to work with some really great people in that field and decided that that was what I was going to stick with. And when I was at University of Arizona, I should also mention that my lab has been a co-run lab for my entire life as an independent scientist. I run my lab with Greg Dusor, who many of you know. He's the chair of the neuroscience department. So we run just one big lab, right? So we got our PhDs in the same place at the same time. So we both were interested in the same kind of thing. It's just been funny that we've had this opportunity to run our labs together all this time. So I decided, along with Greg, when we were still in Arizona, that we wanted to take the discoveries we were making in the lab and spin them out and try to get them into the real world to become therapeutics that would help people one day. And to do that, we thought maybe we could license the technologies we were developing to pharma companies, and then we quickly found out pharma companies don't really do that kind of research anymore. They're more interested in doing the late-stage clinical development. So then we thought we would start a startup company, and we ultimately did that, but then when we would try to get funding for the startup company, we would tell people about the data we had from animal models, and people would say, well, that's cute. What is the evidence that it's going to work in a human? And it's like, well, that's really hard to do in the pain field. And they're like, well, we understand, but if we're going to make the kind of investment that's needed, you have to show us this kind of evidence. And the fact of the matter was, about 10 years ago, very few, if any, people did this in the pain field. So when we came to Dallas, what I decided to do is that I was going to try to fill this gap by really transforming the work that we do. So the slide is up here for a reason. It may not have sounded like I was talking about the slide, but this is an analysis that was done by The Economist magazine, not a scientific magazine, but you probably heard of The Economist. So what they were doing in this analysis was looking at the cost of certain types of health problems, in particular neurological related health problems, and then plotting that as a function of the amount of investment coming from the private sector, mostly from venture capital firms and institutional investors. And what the graph shows here is that pain is a huge medical problem that costs a giant amount of money. The estimates are between $500 and $600 billion a year in lost productivity and overall healthcare costs. But the investment in developing new pain therapeutics from the private sector is almost zero, actually. So what is the reason for that? I think that the reason really is that there's so little confidence in the animal model work that is done and that it'll translate into effective therapeutics. And in fact, we have a lot of evidence now that the animal models, the targets that have been identified and drugs that have been developed in animal models, they have failed over and over again when they've gone into the clinic. The investment is not coming because the kind of data that I'm going to tell you about today hasn't existed. So I can't tell you that I am alone in driving this change. There have been a number of other centers that have been involved in this. And we also have had a huge amount of support from NIH. So the HEAL initiative, helping end addiction long-term, was passed by Congress during the first Trump administration. I think it was in 2018. And this really kind of transformed pain research. And then eventually, NIH created what's called the Precision Human Pain Network. And there's 4 centers across the United States that are part of this funding initiative. and we're moving into our 4th year of this funding initiative. There's a center at UTD, which we lead. There's a center at WashU in St. Louis, a center at Harvard University, and a center at University of Pennsylvania. So there are these 4 big groups that are running this study. So UTD is in pretty good company, I think, for the other universities that are part of this. All right, so the next slide, I'm going to show you a human spinal cord, like the real thing. So if you're queasy, don't look at the screens on the next slide, all right? So now gets into how do we transform our lab from doing what we were doing before. All right, so here comes the slide. So the key thing for us was to develop this collaboration with the Southwest Transplant Alliance. How many of y'all have heard of the Southwest Transplant Alliance? One person. Oh, 3, great. The 4. The Southwest Transplant Alliance is an incredible organization. They do all of the organ procurement for the DFW area and many other areas across Texas, and they are saving lives every day by taking the organs from organ donors and allocating them to people that need them across the state and across the country. And what we have done over the last 7 years or so is to develop this amazing collaboration with them where as soon as the surgeons that we work with are done recovering the organs from the organ donor, they open, so that the body is open from like here to here, okay? And there are no organs inside the body anymore. So if you walk into the operating room at that point, which we do about 2 or 3 days a week with the STA, you're basically looking right at the spinal column, okay, and the ribs. So what we do is we saw the spinal cord open using a specialized saw, and then the ventral side of the spinal column just comes off, okay? And then what you're doing is you're looking at that, but it's still inside the body. And then, so the long thing down the middle is the spinal cord, and the things off to the sides are the dorsal root ganglia. So the dorsal root ganglia is where the nerves that innervate your entire body are found. So in these organ donors, these tissues are still alive. So the donors are brain dead, okay? But their spinal cord is usually still alive. The peripheral neurons are always still alive. And essentially what we've done is we've built these systems to be able to profile the human peripheral nervous system and spinal cord with incredible molecular precision using all these amazing omics technologies that are available now. And we've really taken our understanding of the human nervous system at this kind of single cell level from almost nothing to a really, really detailed view, including how it changes in people that have chronic pain states over the last 7 or so years. So what I'm going to do for the rest of the talk is kind of take you through some of the things that we've learned about the dorsal root ganglion and then also about the spinal cord, but let me first tell you why I'm going to tell you about the dorsal root ganglion and the spinal cord, okay? So I saw Sandy ask how many people have pain, and a fair number of you raised your hands. I have pain almost every day because I have osteoarthritis in both of my knees, and basketball doesn't make it any better, but I love playing basketball. So the reason that you have pain in almost every case is because neurons in your peripheral nervous system, which are found in the dorsal root ganglia, they're at every vertebral level along your spine, and they innervate your entire body, they get activated. You have specialized cells in the dorsal root ganglion called nociceptors. They're the ones that are involved in detecting painful signals from your entire body. They send a signal into your spinal cord, and then that signal eventually makes its way up to your cortex where pain perception ultimately happens, okay? So you have to have a cortex to have pain. There's no question about that. But if you take away that peripheral signal in individuals that have pain, in almost every single one of them, the pain will go away almost immediately. We know that from anesthesiologists doing what's called regional anesthesia, where they give people a nerve block. And if they can localize where the pain comes from, it's like magic, right? You've maybe experienced this. It's incredible. The pain goes away almost instantaneously. So unfortunately, we can't give nerve blocks to people permanently. So what we're trying to do is to understand how these cells change in people who have chronic pain disorders so we can treat them. And then the reason we want to target the spinal cord is because that's the first kind of relay center that sends the pain signal on up to your cortex. So we view these from a drug discovery perspective as the kind of simplest targets that we could hit to decrease pain. Your cortex is a very, very complicated tissue. And hitting it with a drug that's just going to affect pain and not other things is going to be very difficult. I think it's probably possible, but I don't really know how to get there. Modulating the DRG in the spinal cord is a relatively simpler problem, and I think we could probably make a lot of progress on that. So the first thing that we did when we were doing this work was to just simply do the single cell profiling of the human dorsal root ganglion. So if you're really interested in the depth of the science here, this was published in Science Translational Medicine in 2022, and you can read the paper, but what I'm going to do now is just give you a very brief kind of overview of the findings. So first of all, we used a technique called spatial transcriptomics. This is a kind of magical sounding technique that allows you to cut a tissue section, put it on a slide. The slide has these barcodes on them that are 50 microns in diameter, and you can make sequencing libraries from these. And what you can do is you can actually tell the transcriptome of all these neurons, because they're about 50 to 100 microns in diameter at a single cell resolution. So in this paper that we already published, what we were able to do was to identify the entire transcriptome of all the different subtypes of sensory neurons in the human DRG. So we went from really knowing nothing about this to having an essentially complete picture of what these are like in a normal individual in one paper. So Diana Tavares Ferreira led this work in my lab, and she's now a faculty member in neuroscience. She was a postdoc with me when we started this. So if you really want to know about these, you can read the paper, but I just want to point out one thing about the data that I think is really interesting, and that I hope you also find interesting. So many people who have chronic pain also have comorbid immunological disease. So that's been a really big mystery. Like, why does chronic pain, especially neuropathic pain, cause people to have immunological problems? So what we discovered when we were doing this analysis is that each particular subtype of sensory neuron expresses neurotransmitters, neuropeptides, that if you look at immune cells, what you'll find is that there are receptors for all these individual neuropeptides in immune cells. And you find these immune cells in your joints, in your skin, all over your body, right? So if you kind of map the individual cell types, some of them have the ability to communicate with monocytes or macrophages. Some of them have the ability to communicate with T cells. And we feel pretty confident this is kind of driving these immune changes that happen in individuals. And the thing that's also very interesting about this is that if you look in rats and mice, which is what many of my colleagues in the field use, and you look at these the diversity of these neuropeptides, they really just have two of them, CGRP and substance P. But if you look in humans, they have about a dozen of them, and they have a very cell type specificity to them. So this complexity you just wouldn't be able to appreciate from doing animal studies. So as we got deeper into the work that we were doing on all these DRGs from these organ donors, we wanted to address the question, what's happening in the nervous system of people who have painful neuropathies? So we can't choose the organ donors, obviously, right? The people die of a tragic accident, and we get the tissue, and they are at whatever stage of their life they were at when we get that tissue, right? So what we discovered after doing about 50 recoveries is that we were getting a decent number of individuals who had diabetes and diabetic neuropathy. And if you know much about the population here, that probably doesn't surprise you very much in Texas. So over the 7 years we've been doing this project, we've now done recoveries from about 400 organ donors. So we have a very large tissue bank. And Stephanie Shires, who is a research scientist working in my group, she's been responsible for doing all the quality control on all these tissues. So basically she's been doing basic histology on them all, okay? So one day she comes into my office and she says, I think I've found something really interesting. I'm seeing these pathological structures within the DRGs. They're tiny. They look like accumulations of nuclei, but they're in areas where I know there should be neurons, and there's not neurons anymore. There's just these accumulations of nuclei. There's some kind of cells. And I went through all of the data that we have, and I found that I only see them in people who had diabetic neuropathy. Turns out there's actually a couple other places that we see them, and I'm like, oh, that's really interesting, because this could be a pathology that, is linked to diabetic neuropathic pain. So I asked her what they were, and she's like, well, I looked in the literature, and I didn't really find anything in the literature. There's nothing in the models in rats or mice of diabetic neuropathy that people have found, but eventually I found this book written by a French pathologist in French in the 1920s. So this book is by a pathologist named Jean Naget, and what Jean Naget was doing amputations in rabbits. So this is something you would never do nowadays, and you would never get approval for it, probably rightfully so. So he was doing these amputations in rabbits and then studying how the dorsal root ganglia was changing in the rabbits, and he found these things that he described as Naget nodules. So basically what they are, they're these accumulations of nuclei, and then the neurons that are near them sprout in and they form these pathological looking structures. So, I want to mention one more thing about this. So these structures look like neuromas. So people who have an amputation or have a traumatic nerve injury often get a neuroma. And essentially what a neuroma is when your peripheral nervous system can regrow, right? But if the peripheral nervous system regrows unproductively because you had an amputation and it can't go anywhere because of the stump, these neuromas form, and they basically become like intertwined axons that are just growing unproductively, and they become sites of ectopic activity. So these people often have pretty severe neuropathic pain, but if you inject local anesthetic into the neuromas, the pain goes away. And so people often have these neuromas resected. And what she's seeing in these DRGs is these tiny, tiny neuromas just all over the DRGs. So she did an amazing job of kind of classifying all this, and this paper has now been accepted in Nature Communications. It's supposed to come out in about 2 weeks. So what she found was that the severity of these Naget nodules in the DRGs in people with diabetic neuropathy is very extensive, number one. Number two, you could ask how many DRGs are affected, how many neurons are affected by this? And these are dying neurons, right? So this is neuronal loss within the DRG. It's not a small number. It's between 30 and 40% depending on the individual. So it's a large number of neurons that are dying within these diabetic DRGs. And then we don't have a large sample size for other kind of pathologies, but what we found was that we do see it in some other instances. We had an individual who had a painful alcohol-induced neuropathy, had another individual who was diagnosed with fibromyalgia, but we're pretty sure they had an autoimmune neuropathy. And we see it in those people with neuropathic pain, but we don't see it in rheumatoid arthritis or osteoarthritis. So it seems to be really exclusively happening in people who have neuropathic pain. So in the 1920s, Jean Naget didn't have the imaging or sequencing technologies that we have nowadays. So what Stephanie did was to really go into detail about trying to understand what's happening within these tiny little neuromas. So there's a very famous neurologist named Steve Waxman at Yale who studied voltage-gated sodium channels and how they contribute to pain for many, many years. And he's done this amazing work looking at the neuromas of people who have neuropathic pain. So these are now at the stump of amputation, okay? And these neuromas are totally full of a sodium channel called Nav1.7. So our hypothesis was that if these are really little neuromas, they should be totally full of Nav1.7. And that's exactly what you see. So here's the Nav1.7 immunostaining. They're just totally lit up with Nav1.7. And they also have these little plaques. So these are accumulations of the sodium channel. It's likely these are sites of spontaneous activity generation. So, this would cause the burning and shooting pain that diabetic neuropathic pain patients so often have. And they look exactly like the neuromas that you would see out in the periphery. It's just they're maybe one one thousandth of the size. But these human DRGs are just totally full of these little pathological structures. And then we can use really advanced tracing methods to trace single axons. So what Stephanie did was to look where they're coming from, and it turns out that they're coming from nociceptors, so these pain-detecting neurons that are near the neurons that are dying. And we could see it over and over again. And they just form these amazing structures. And if any of y'all are a neuroscience aficionado, you probably know that DRG neurons are so-called pseudo-unipolar neurons, right? They have one axon that comes off. We see in the people with diabetic neuropathy that the neurons actually turn into multipolar neurons, which has never really been described before. So what it means for their excitability, we don't know exactly, but we're pretty sure they should be producing lots of action potentials, which would cause the burning and shooting pain that people with diabetic neuropathy have. So this picture here just kind of shows how extensive it is. So if you can see this well, this is what a non-diabetic DRG looks like. It has a very nice organization looking to it, and it's just like pure chaos in the diabetic neuropathy DRGs. So the next question we had is, what are the neurons that are dying? How is it just random neuronal death? Is it a particular subset of neuron that's dying? So Aish, who's a postdoc in my lab, led these studies. So what she did was to take the same DRGs and do single nucleus RNA sequencing. So this is another omics technology, and it allows us to look at the transcriptome of each individual cell within the DRG. So it's really, really cool technology. And what she found when she did this, so on this side is the non-diabetic neuropathy UMAP, as we call it. This side is the diabetic neuropathy UMAP, and then you can see the circles. Those are clusters of cells that are only represented in the normal DRG. They're not there at all in the diabetic DRG. So presumably these are the cells that are dying, okay? So we're losing these populations. So it turns out that the cells that are dying are so-called low-threshold mechanoreceptors. These are your light touch neurons. So this was another kind of eureka moment, because it's exactly what happens in people with diabetic neuropathy. I see Jane's nodding her head, so we must be on the right track, right? So people with diabetic neuropathy usually report, the first symptom is they start to have loss of light touch and proprioceptive issues, often accompanied by falling. And then pain seems to start later on. And then oftentimes what happens is people become completely insensate. They don't have any sensation in their legs, sometimes their hands. And the pain sometimes does eventually go away. But there are these very complex neuropathies with gain and loss of function, and the pathology we've seen explains it perfectly. It's just not the same as what people have thought, where it's the axons dying back in your leg. It's actually happening within the DRG. And the cells, it's not the axons that are dying, it's the entire cell that's dying. So the issue with the low threshold mechanoreceptors dying would cause the loss of light touch and proprioception, and the pain would be driven by the sprouting of all the neurons forming the Neget nodules. So ultimately, I think this study has led to a completely different understanding of what diabetic neuropathy actually is. It's a neurodegenerative disorder. And we need to think about treating diabetic neuropathy in a totally different way. We need to not really be so concerned with thinking about how we're going to decrease the pain early in disease. We need to treat with neuroprotective agents, because ultimately it's the death of the low threshold mechanoreceptors within the DRG that are driving the pathology. So a big thing that we're trying to do now is to better understand what's happening to these low threshold mechanoreceptors within the DRG at early stages of the disease, and we have a little bit of data that suggests it's a programmed cell death, caspase-3 driven phenomenon, and that's good because we can manipulate that pharmacologically. So it could offer some really great opportunities for much better treatment of the disease. Okay. So what I'm going to do now is transition to telling you about the spinal cord work. So this work has been led by Catherine Gabriel, who's right there, and Olivia Davis, whose work I'll tell you about on the next slides. So the spinal cord work we're doing to understand the molecular architecture of the human spinal cord, because we don't really know very much about it. And then the other big objective that we have is to try to understand pain mechanisms in the CNS, and to understand if we can find ways to find what are called the projection neurons. So these are the neurons that receive the nociceptive input in the dorsal horn of your spinal cord and send the signal on to the brain, okay? You can't have pain without that signal getting out of the spinal cord and up to your brain. So if we can sequence all these neurons and understand all the genes they express, potentially we can come up with a way to silence these neurons and stop the pain signal from ever leaving the spinal cord. And these really cool looking images here are what's called xenium spatial transcriptomics. So the first technique that we did spatial transcriptomics on about 5 years ago was called Visium spatial transcriptomics. It had a resolution of 50 microns, so we could see like one whole neuron. This technology has a resolution of 200 nanometers. It's incredible, right? So the advancements that are happening in transcriptomics right now are just absolutely crazy. It's a really fun time to be doing this kind of work. So we have a Xenium machine up at the main campus at UTD in the imaging core. And I'm lucky enough to be the director of the imaging core up there. So slightly different kind of imaging than you guys do down here. But so you can see where all these different cells are within the spinal cord. So our workflow for this, which was developed by Catherine and Olivia, is to recover the spinal cords from the organ donors and then do single cell and spatial transcriptomics on them. And the goal, of course, is to do all the stuff that I was just telling you about. So we've been able to identify all the different cell types in the spinal cord and their transcriptomes, and I'm going to just tell you two pretty quick stories about two cell types. So the first one I'm going to tell you about is about astrocytes. Astrocytes are pretty important for lots of different reasons. For people that do brain imaging, they're really important for modulating the bold signal, which many of you do, because they ultimately link the neuronal activity to the blood flow within the brain. And then we think they're pretty important in the spinal cord because they become activated after nerve injury and may communicate pathology to the neurons. So we haven't really gotten into the chronic pain spinal cords like we have on the DRGs, but we do have some interesting findings on the astrocytes. And the key finding is that there's just a huge sex difference in the astrocytes and the human spinal cord. So we don't see sex differences in most of the other cell populations, but in the astrocytes, we really do. And what we find is that many of these are profoundly different between the males and the females, with a huge bias towards the males. So it seems like there are at least 5 populations, maybe more, that are enriched in the males, and we just don't see these populations in the female spinal cord at all. And if you look at the signaling pathways that are enriched in these male-enriched astrocytes, what we find is that they're involved in the integrated stress response, or ER stress, and also interferon signaling. So the reason that I'm highlighting this is because there's this idea from the preclinical literature that spinal interferons decrease pain signaling. And we only see this in the males, and we think it potentially is a cell type specific driven pain dampening mechanism that is only present in the males. We haven't found any such mechanism in females, but it's possible that as we do more samples, we'll eventually be able to find such a mechanism that might exist in the female spinal cord. And it's not just the transcriptome of these cells that looks different. The cells actually have much different morphology between the males and the females. So astrocytes are supposed to look something like that picture over there. They have a small cell body, and they have all these extensions that come off them, and they have end feet that are around the vasculature. So this is a xenium transcriptomic image, and what it is an astrocyte-specific marker. So you can see how amazing the resolution is on this stuff. It's really remarkable. But the thing is that only the females look like what an astrocyte, like what we think an astrocyte should look like. So the males, the astrocytes have like a very enlarged cell body, and they have very few end feet that seem to be coming off of them. So I don't know why this is, but it really is striking. There's just a huge difference in the morphology of these cells, in addition to their transcriptome between the males and the females. So we're hoping that we can understand this better. All right, so now what I'm going to do is tell you about the neurons. So this is work that Olivia has done. Olivia joined my lab from Glasgow. So she was at University of Glasgow for her PhD. She did her PhD in a guy named Andrew Todd's lab, and Andrew Todd is very well known for being the world's leading spinal cord person. So I was really lucky to recruit her here. She wanted to come specifically to do this project, and she's done a really great job of kind of making it happen. So what we did is to do, again, single nucleus sequencing on all of these spinal cords, and we have a very large sample, about 12,000 cells that we could do this clustering on, and we identified inhibitory interneurons, excitatory neurons, and also cholinergic population of neurons in the dorsal horn. And what I'm going to do is just tell you a little bit about the excitatory neurons, and then tell you about how we're going to get to finding these projection neurons. Okay, so the projection neurons, again, are the ones that send the signal out of the spinal cord up into the brain. So first, when you look at the different populations of the excitatory neurons, we find that there are about 8 of them, and none of them appear on the surface to be projection neurons, which was a little bit disappointing, but I'll tell you how we might get there on the projection neurons. So when we look at where these are in space, we could see that some of them are just found in the superficial dorsal horn, like this population and that population over there. Some of them are kind of spread throughout. If they were projection neurons, what we would expect is that they would be like in this area here and in this area down here. So this is also kind of disappointing because we didn't see any particular population that was only in those areas. But we did have a pretty nice kind of sanity check here because pain and itch both get processed in your spinal cord. So chronic itch is no fun either, but it's a little bit simpler than pain, okay? There aren't that many different kinds of itch. I mean, there's kind of just itch and then there's itch, right? So, and we know a lot about the itch circuit from studies in mice. And Olivia analyzed this in quite a bit of detail, and we can find the itch circuit in humans very easily. So in mice, there are these cells that express this peptide, GRP. It's the gastrin-releasing peptide, and also the receptor for the peptide, GRPR. They're 2 separate populations. We find them in the human spinal cord. They're in lamina 1 and lamina 2. This is lamina 1 and lamina 2. They're in exactly the same spot in humans. And then when we pair them up with the sensory neurons that are the itch sensory neurons that we've also identified, we find that the receptors that are expressed by those peripheral neurons match up beautifully to the neurons in lamina 2. So we're pretty sure we've now identified the itch circuit in humans. It's conserved with mice. So this is a case where the work that's been done in mice could lead to really great therapeutics, and GRPR antagonists are actually moving into the clinical development for chronic itch, which people that have chronic itch, it's really a horrid thing, right? So if we can find ways to alleviate that pharmacologically, it'd be really great. And I think we should be able to do that in relatively straightforward fashion. All right, so what about those projection neurons? This will be the last little thing that I'll tell you about before I take questions. So we know about some markers of projection neurons from work in mice, and so the first thing we did is to see if we could find them in any particular population in the human spinal cord. And again, we couldn't find them. So what we decided to do was go back to Andrew Todd's lab. So remember, Andrew Todd is the person that Olivia did her PhD with. So there's a postdoc in Andrew Todd's lab, Andy Bell, who has done this really amazing project in mice to do what's called lineage tracing. So during early development, different kinds of neurons have different kinds of transcription factors that they express, and that those transcription factors ultimately determine their fate, okay? So from these mouse studies, we know that this transcription factor, FOX2A, determines the fate of projection neurons. So you can turn on this gene during early development in mice, and you can do what's called fate mapping, and then you can take those neurons in the adult and you can sequence them all. So that's what Andy did. It's an amazing approach. And we thought, this is how we're going to find the projection neurons in humans. We'll just take all these markers from these, and we'll find the population. And it was an utter and complete failure. So here's one example, ZFHX3. So this marks a very particular population of projection neurons in mice. it's in every population of neurons and excitatory neurons in the human spinal cord. It just does not work, okay? So shucks. How are we going to do this? All right, so we had a couple of ideas. So one is projection neurons, so neurons that have long axons and go a long way in your brain, they're bigger than the inner neurons that just connect within a brain nucleus. This is the same for sensory neurons. They're very big, and it's also the same for spinal projection neurons. They're very large neurons. They have a very long axon. So Olivia thought we can probably just identify them by size. So she went through this crazy exercise of measuring the size of all these neurons in all these experiments, and what she found was that indeed there is about 1% to 2% of neurons in the spinal dorsal horn. That's about the same number of what we think should be projection neurons, and they're bigger than 50 microns. So we probably can find them by size. But the question is, how can we label them developmentally? So people die at all ages and can become an organ donor at every age. So about 3 years ago, we started doing recoveries of nervous system tissues from pediatric organ donor cases. It's a total tragedy, but we try to make the most out of this as we possibly can, learn as much about it as we can. So we started doing developmental studies in the DRGs and spinal cords of these organ donors. And what we have discovered is that this gene that I told you about that specify these neurons in mice is also on in early development in humans, and all of those neurons that express FOX2A in humans are large. Now we've done single-cell sequencing on the pediatric cases and the adult cases, and we know how to identify the projection neurons now. So this developmental study has allowed us to be able to find now these targets. So we haven't actually pinpointed them yet, but it's going to work. It has to work. There's no way it's not going to work. So we built these panels now for the spatial transcriptomics to be able to label these neurons, and we're like running all those experiments now. Once it's done, we should have the identity of those projection neurons, and that will allow us to find the targets that then we can make drugs to stop the ascending pain signal. So on the spinal cord, we have now gone from not really knowing very much about the human spinal cord at all to having a really good atlas of all of the cell types there. We know that there's some pretty important sex differences, in particular in astrocytes and microglia. We know there is some conservation of neuronal subtypes to the mouse, in particular for itch, but in many cases there isn't very good conservation. But thanks to these developmental studies, we can do this fate mapping of these projection neurons, and that's what's going to get us to our next series of drug targets to treat pain by blocking the signal from getting out of the spinal cord and going to the brain. So I want to thank all the really great people that I'm so lucky to work with in CAPS. So the center was founded by myself, Michael Burton, who's right next to me, and Greg Dusor. And it's great to work with them and all the other people that we've now recruited to the center. The work that I told you about today was led by Deanna, who's faculty at UTD now, Stephanie, who's a research scientist in the center, and Catherine and Olivia. And couldn't have done it without the Southwest Transplant Alliance and the organ donors and their families. And every single thing that we did was funded by NIH. And thank you all for listening, and I'll take questions.
I have a question.
Oh, my mom has a question. Yikes.
Okay, so this is, I've heard you talk about this, and we all know where NIH is right now.
Mm-hmm.
So this may be a loaded question. You can answer it if you want to, or you can tell me privately. But how is the current situation with the NIH going to affect you and all of these people and the great work that you're doing? That has been my basic question from the start.
I'm glad you asked that, Mom. All right, so it's been stressful, as I'm sure a lot of the other PIs in this room would agree. a lot of us are thinking about the future and whether we're going to continue to have this funding. I think the Precision Pain Network, for instance, has been extraordinarily successful. I told you about the work that we've done here. There's been equally incredible work that's been done at the other centers. As of right now, I think the chances of the Precision Pain Network continuing are very slim to none, because they're not going to keep doing U19s. It's a mechanism that I understand that NIH is going to eliminate. So another thing I can tell you is that Olivia, who's amazing, has decided to go back to the UK. So she's leaving in August. We'll finish her work here. She's going to be amazing where she is, but you know, we used to always keep these people in the United States. So it has been tough. It's been tough for everybody, though, not just us.
Yeah, and so that's why the public, we're going to need your funding even more during this time to make sure that we don't stop and these networks don't stop. So I think that's where we go back to the public and say, there's more urgent need to fund this basic research because if everyone has pain, so I think we've got a bigger message publicly to get support so it will continue because it can continue. So we've got to make that our
I guess I have one other comment to follow on that. In our lab, we've spun 7 companies out of the lab. We plan, like every time we find a target, we spin a company out and we start doing drug development around the target. So we're also very interested in people that are interested in investing in the mission. So we've had two companies now that have developed molecules that have gone into clinical trials. I think that any kind of early stage investment like this is risky, but we've had a good history of success, and we really want to change the way pain is treated. There was a new drug approved for pain early this year, suzetrigine, the NaV1.8 inhibitor. That targets nociceptors. It works for acute pain. It also seems to work for diabetic neuropathy. I think it works because of all the stuff I told you about today. So there's a lot of opportunities to have a big impact on this particular field right now.
So I want to echo Sandy's comments. On LinkedIn, I see a lot of us talking to each other, and that doesn't do us a lot of good. We really need to talk to the public. And so those of us who are able should be talking to our newspapers, our media outlets. So it's just a comment.
Yeah, totally agree with that too. Great point. Well, I mean, we started writing op-eds about 2 months ago. None of them have been published yet, although I think one is maybe getting close. But you know, you have to also get over the hump to actually get them published, which is a bit of a mystery to me how exactly you make that happen.
So I'm going to ask a question back to the science. Like, I'm kind of surprised, I guess maybe I'm naive, to think that itch and pain are the same. You know, I think about itch being, you know, like some type of seborrhea or something, and it's more external, but it's triggered by the same thing. But to say itch is the kind of carried by the same system is like, whoa.
Yeah.
And if you block that to the brain, do you do inappropriate things?
That's a good question. So I don't think so, but it is possible. You know, the idea of the itch system is that it evolved to make sure that you keep pathogens off of your skin. Mm-hmm. So the itch nociceptors we call them nociceptors because they're C fibers, they're non-myelinated, they have free nerve endings. They're a subset of neurons that innervates the outermost layer of your skin. So they're really there to detect stuff that's crawling on your skin, basically, right? And then what do? You basically wipe it off. But when those neurons get activated inappropriately, like by histamine or things like that, it causes this itch sensation. And then you scratch because this circuit gets shut off by another subset of neurons. So in mice, we understand the details of that circuit with incredible precision. And I think from what we've seen here, the circuit is exactly the same in humans. So,
So I have an online question. Is your research relevant for neuropathies other than diabetic neuropathy?
yeah, so I think we've seen that there are other neuropathies that have this neurodegeneration, but we know clinically that not all neuropathies show this kind of mix of loss of light touch and proprioception and pain. So we spend a lot of time also just trying to understand how the excitability of human DRG neurons change in people that have neuropathies. So we know that the neurons start to develop what we call spontaneous activity, and that seems to be driven by a mix of changes in sodium channels and intracellular signaling pathways that we can block pharmacologically, including by one of the molecules that we developed, or targets we identified in our labs that's in phase 1 clinical trials now with the company that I started with my brother. So hopefully that'll be really successful when the company's called 4E Therapeutics, but hopefully that'll be a very successful strategy for these kind of less neurodegenerative neuropathies.
Are there any other questions? There's some. Let me ask another question online, and then there's some more out there. Any way this research can help reduction in depending on pain medications for diabetics and arthritis?
So one of the things that we're trying to do is to make a transition from opioid-based therapeutics to non-opioid, more efficacious therapeutics, and also to have therapeutics that are not really so much used palliatively, but can cause disease modification. So I think at the end of the day, pain happens because of some kind of pathology in most people, and our goal is to understand what that pathology is and try to develop therapeutics that will reverse it. So ultimately, if that works, people hopefully wouldn't have to take those drugs for the rest of their life. Like they're often stuck with drugs like opioids or gabapentinoids and things like that now.
Ted, thank you for the really illuminating talk. It sounds as if a lot of these innovative developments that you've been able to study were made possible maybe by the resolution changes. So you had alluded to how dramatic the difference in resolution with some of these methodologies has become. And I just was curious, given where we are now versus where we might be in, say, 5 years, is there anything, do you think it will really revolutionize this area? Because it seems as if you're getting into really core mechanisms that just simply never would have been possible to observe before. Are we at the sort of the gateway toward some much greater progress?
Yeah, I think so. So I think that the single biggest thing that's going to come that's going to really change the whole game is, right now we're doing transcriptomics, right? So we're measuring RNA. So RNA is important, obviously, but protein is ultimately what does the business. So the RNA gets translated into protein, but we can't measure the protein with the kind of spatial resolution or specificity that we can the RNA. So spatial proteomics is advancing really quickly, and when that gets to the point where it looks like the spatial transcriptomics now, that'll totally change our ability to identify targets, because there won't really be guessing anymore. We'll be looking directly at the proteins that we think are involved in causing the pathology. So I think that's the biggest area where we have a little bit of a lack in the technology, but all these technologies are advancing so fast right now. It's just incredible. Yeah.
Thank you. My question is going back to opioids. Is there an opportunity for the development of a drug that is just as effective but without the risk of addiction?
Yeah, I think that's like suzetrigine, for instance. So that's the NaV1.8 inhibitor that just got approved for acute pain. The clinical trials showed that it was as effective as opioids actually for acute pain. So, and it's not addictive. The target's not expressed in the brain at all. So opioids are addicted because they're all over the reward circuit. So for actual clinical experience, will suzetrigine actually be as effective as opioids? I don't know that will ultimately be true, but it's being prescribed all over the country now for acute pain. So the suzetrigine. So this was the NaV1.8 inhibitor that was developed by Vertex. Is Journavax is the trade name for acute pain, acute post-surgical pain. Yeah, specifically. But they're going to go for approval for diabetic neuropathy this year. They had a negative clinical trial that they spun as positive for radiculopathy. I can't see how they're going to go for approval for that. But their diabetic neuropathy phase 2 data look great, and they're doing, they finished phase 3 actually. So I think they're going to go for approval for diabetic neuropathy this year. And then Merck has a NaV1.8 inhibitor that's somewhere in late development. Latigo, which is based in LA, they have one, Site 1 has one. So I mean, it's going to be like the CGRP therapeutics for migraine. There's going to be tons of NaV1.8 inhibitors that go towards approval in the next like 2 or 3 years. So those could be as effective as opioids in theory, with no addiction potential.
I'm not sure. This is the same question, but what's the name of the new drug invented for diabetic neuropathy? Is that?
So that was the one I was just talking
That's
about,
what
but it's
I
not approved for diabetic neuropathy yet. So it's finished phase 3, but they haven't gone for approval.
And then there was a question about what causes change to the astrocytoma?
Oh, what causes the differences in the astrocytes? I wish I knew. We don't know. This is like one of these mysteries. The beauty of getting all this data is like we know the active genome of these cells. Presumably it's something related to interferons, because that's like the big difference that we see. But we haven't been able to do experiments to see like what is it that the interferons are doing to these astrocytes. But I could also, my hypotheses are usually wrong. So I mean.
Well, what do you do for your pain from basketball?
I play more basketball. Just keep active is what I do.
So you don't give in, you don't take anything?
I have a very close physician friend. So me, Greg, and this guy, Amol Patwardhan, he's an anesthesiologist at UT Southwestern. We all did our PhDs in the same lab in the same time. I was with Amol yesterday. And I told him he needs to prescribe me some Journavax because I'm dying to try it and go play basketball with my NaV1.8 channels blocked, because I think I'll be like I'm 20 years old again.
You're going to be your own individual
yes, totally.
test case.
I can't wait.
Wow. Well, thank you so much,
you,
Dr.
Sandy.
Price. That was amazing, amazing talk. Thank you. Thank you. I know that everyone's going to rally around and ask their questions now, but I did want to ask you about Ted's pain cream. Well, it's gone, but what did it do? What was Ted's pain cream?
It had resveratrol and there was one with resveratrol and lidocaine, one with resveratrol and aspirin, basically. And the resveratrol is amazing for inhibiting nociceptors. You just can't take it orally because it doesn't ever get out of your gut. So it actually absorbs across the skin pretty well. But unfortunately,
can we develop it again and pass it out or sell it?
Tell Greg that we need a Greg's pain cream.
Great. I know. I saw that
there's
commercial.
There's a commercial about it.
I know.
Right? Yeah.
I love that commercial. Well, thank you. Thank you. I think you can see one of the questions we want to ask together is, what is the impact of pain on brain health? We know when people are experiencing pain that they change, and so won't this be a great collaboration between our two centers? And we would be so honored to work with Dr. Price, Dr. Duser, and Michael Burton. So thank you for a brilliant talk today. Thank you all for joining us. We look forward to seeing you again April 25th. Our guest speaker is going to be Dr. Paul Zak. Dr. Zak will define emotional fitness to share research that has established the 4 Ps for emotional health: personalized, preventive, predictive, and participatory, to describe the holistic approach to mental well-being. Well, that's a big part of what we measure in our Brain Health Index, so we can hardly wait to see Dr. Paul Zak. For those of you that are out there that don't know about our series, The Brain and Owner's Guide, I hope you will, not, sorry, the Brain and Neuroscience guy, that used to be the old name. BrainHealth Presents, sorry, I went back to old times. BrainHealth Presents is coming. We have one of the world's expert on memory. Fascinating. It's one of the most provocative thoughts about memory is coming up next Thursday. Sign up for this series. There's cards out there. Go on our website. Brain Health Presents. You won't want to miss it. All 4 speakers coming throughout the year are incredible. Sign up for your sleep challenge if you haven't already, because our sleep matters. Dr. Price actually helped us recruit Dr. Matt Walker that will jointly be in neuroscience, work with brain health, but also bioengineering. So we're really lucky to have him come on board. So we will see you in a couple weeks. Thanks for joining us.